And our first speaker for this session, I'm super excited to introduce Dr. Barbara Zdrazil. Barbara is a scientific leader with 15 years of experience in cheminformatics and computational drug discovery. With an academic track record and international reputation, she currently works as ChEMBL Group Coordinator at the European Bioinformatics Institute, EMBL-EBI in Hinxton, Cambridge, UK. Her current scientific focus is to aid in the delivery of AI-ready small molecule drug discovery data, lower the barriers to efficient data usage globally, and automate data extraction and curation processes without loss in data quality. And today her presentation is entitled 15 Years of ChEMBL Challenges and Opportunities. To you, the floor is yours, Barbara. I'll let you share your slides.
Thank you, Sarine. Can you see my screen?
Yes, fantastic.
Great. And can you hear me well as well?
Yes, we can hear you really well.
That's good. Thanks a lot for the kind introduction and also for the opportunity to speak here today. So thanks a lot to the organizers for inviting me. So I would like to talk to you about ChEMBL, a drug discovery database that hopefully most of you have heard about or even have used their data. So at the occasion of ChEMBL's 15th anniversary, we very recently had a symposium on campus, a 2-day symposium, and I just wanted to share a few pictures. So of some former and current group members, you can see on the upper part of the presentation. We also had a nice cake and a lot of people attended. So that was really a nice thing to do. And before I start talking about ChEMBL, I just wanted to briefly introduce our institute. So I work at the EMBL-EBI, so the European Bioinformatics Institute in Hinxton, UK, close to Cambridge. But this is actually part of a larger European infrastructure of the European Molecular Biology Laboratories. So there are 6 different sites that you can see here on the screen, and we are basically funded largely by the 20 member states of EMBL. And the EBI, where I work, we host open resources for the life sciences, so a focus on big data. And here you can see them on the slide, grouped by different topics. And we are really, so ChEMBL is in the sector of chemicals and small molecules, like as well CHEBI, which we also host in our team, the chemical entities of biological interests database, ChEMBL for patents, and then also UniChem. And there are a lot of other resources, and there has been a mention of this already in Marta Head's talk. So AlphaFold database, for instance, that you might have heard of, then the Protein Data Bank in Europe. And I just wanted to mention that even though most of these data sources don't focus on small molecules, they still would reference or include a lot of small molecule data often. And just today, we, for instance, released the first data release of a project called BioChemGraph that links the PDB-E structures together with ChEMBL bioactivity data and the Cambridge structural database data. So watch out for that. And just as a side note, our campus is growing, so there is really a focus to increase and expand these bio industries, and it will hopefully then be one of the world's highest concentrations of expertise in genomics and computational biology. So that's quite exciting, I think. And now let me switch to ChEMBL, my favorite topic here. So ChEMBL is, as I said, a database which is now quite mature because it reached its 15 years this October. And what is interesting to note is that over time, ChEMBL has grown a lot in size, and we have heard that from Marta before. So data quantity is, of course, important, but it also has grown a lot in terms of its content, and then also in terms of the complexity of the data that we host. And this also has an effect on the complexity of the data schema, as you can see on that slide. So initially, ChEMBL, really, when it was launched 15 years back, this was a database that focused on data extracted from the medicinal chemistry literature. So structure-activity relationship data, so congeneric series of molecules, along with the bioactivity data and the assay details. And you can see here in the middle of the slide, ChEMBL version 1 was a very tiny database schema, a few tables. Now we expose around 80 different tables to the public. That, of course, means we also need to curate the data. So the whole curation process takes a bit longer, is more tedious, but we hope we still would keep the good quality that ChEMBL is known for. And what you can also see on this slide is that how the data content has basically changed. So on this graph in the bottom left, you can see how the share of bioactivities from the different main sources in ChEMBL, which is publications versus donated data sets, has changed over time. So at the moment, we reached a point where more than half of the data, the bioactivity data in ChEMBL, is not actually from medicinal chemistry literature, but has been donated by data donations from dedicated projects and so on. And of course, this changes the way we need to think about the data, right? So we needed to a bit adjust our data deposition process. And also, as I said before, the data is now more diverse and more complex, which influences our curation strategy. So on the left-hand side, you can see all the different sources, so the main high-level sources of information that data is being deposited into ChEMBL. I mentioned scientific literature, deposited data sets. There is some patent data as well in ChEMBL, so bioactivity data extracted from patents. So we used our SureChEMBL database for that. But this is rather small compared to the whole size of SureChEMBL. So it's around 186,000 different compounds. The way we identified those patents that we wanted to integrate data from, along with the bioactivity values, was identified in a project with the Illuminating the Druggable Genome project. So this was really a focus here on understudied targets. Then also we have some data from some public databases, so other databases like BindingDB. So some of their patent data has made it into ChEMBL. And we had twice, I think, a deposition from a confirmatory assays from PubChem. But this isn't something we are doing regularly, so not really on a regular basis. And then the other thing that I wanted to mention is we also do quite extensively curate drug and clinical candidate information in ChEMBL. So this is drug data that's not stored along with the bioactivity data, but is extracted from a variety of sources that I will explain in a minute. And by integrating all these different data sources into ChEMBL, what we in the end can say is that basically the bioactivity data in ChEMBL covers all the different stages of this drug discovery and development process. So really from target discovery to lead discovery, lead optimization, preclinical development, and also the clinical phases. But in terms of quantity, most of the data is really covering more this preclinical stage. And because I'm not sure that everybody's so aware of this drug and clinical candidate data in ChEMBL, I wanted to make a point here that this is a quite tedious process. So we take data from a variety of sources that you can see on the left. So for instance, FDA Orange Book for approved drugs, clinical trials, use and applications for clinical compounds. And then we supplement those with additional sources like DailyMed for indications. So we collect all that information, we curate it, and we integrate it. And then we do a few different annotations. So on one hand, we have to often map the name of the chemical to a structure in ChEMBL in the database. So often those chemical structures are not provided. We then annotate the mechanism of action target. We also annotate the indication or disease that this drug was intended for. And we also provide drug warning information. So black box warning or when a drug has been withdrawn. What is new since the last release of ChEMBL, which is ChEMBL 34, we now included drugs from the European Medicines Agency as well. And this data then goes, so first it's being collected in a separate database instance that we internally call DrugBase, but then it makes its way into ChEMBL once a year approximately. And from ChEMBL, we also then basically donate this data to Open Targets. Open Targets is another platform that is maintained on campus at the EBI. It's a platform for linking genes to diseases, and they have different sorts of evidence for these links, and drug data is one of them. So from drugs, I wanted to go to natural products. I think there's also quite a hot topic these days. And so we wanted to make an effort in these past 2 years that I joined the ChEMBL team to better flag natural products in ChEMBL. So what we did is first we have updated our natural product likeness score. So before that, we had an algorithm that would only flag drugs if they were natural products, and it was a fragment-based approach. And now we updated this to the algorithm published by Peter Ertel some years ago already, and we took an RDKit implementation by Greg Landrum that uses fully open data sets for its calculation. So that's available. And then one release later in ChEMBL 33, we also introduced a natural products flag. So now we are flagging all molecules that would map to a molecule in the coconut database as a natural product in ChEMBL. What I then did when we had this natural product likeness score first, I wanted to analyze this distribution in the ChEMBL database. And what was nice to see is that this distribution basically reflects what has been reported by Peter Ertel in his original paper. So the average value is around -1, which is somewhere between drugs and synthetic compounds. And this is basically what ChEMBL reflects. Also interesting, if you look at the distribution of the score across different journals, so in this right bottom corner, you see it's approximately the same in different journals, but Journal of Natural Products, on average, the molecules published in this journal have a higher natural product likeness score, which isn't surprising either. And then I went and I just collected all the drugs, approved drugs reported in ChEMBL, along with the first approval date, and plotted the natural product likeness score. And what you can see is that there is a slight decrease in natural product likeness since like kind of 20 years, no, 10 years, maybe it's slightly going down. And this sparked our interest in this topic of natural product likeness and drugs and drug likeness. So we started a collaboration with Herbert Waldmann from Dortmund University, and he's also working at the Max Planck Institute. He's an organic chemist interested in natural products, and he developed a concept, or he published on that is called pseudo-natural products. And what this is basically natural product fragments combined in new ways that are not that can't be synthesized by nature. So basically you keep all the beneficial properties of natural products, but you would be able to reach out to new chemical space. So this was a collaboration then also with Paul Leeson, who was interested in analyzing natural product likeness and pseudo-natural product likeness in the ChEMBL database. And what we did is we compared clinical compounds versus background compounds, so all the other compounds that aren't marked as clinical compounds, so clinical candidates or drugs in ChEMBL, because we wanted to see, do we see an enrichment in pseudo-natural products over time? So is this an increasing popular concept? So do we see kind of a natural selection in small molecule drug discovery? And as you can see, we saw this enrichment. If you want to know more about this, we recently published this, so please go and read our article, and we're happy to answer questions about this. But in the framework of this collaboration, we also published a reliable and robust data set for community use. So to us, it's because we work in this environment where we make everything openly available. We are also interested to make our software available and also to create those data sets for community use. And the data set we used for this analysis is a data set of compound target pairs that helps you explore differences between drugs, clinical candidates, and other bioactive compounds. The paper came out, I think, 2 days ago, and it describes how this data set has been established. And we also provide all the code on GitHub, the data sets on Zenodo for different ChEMBL releases. And we are also now making this available with every new release of ChEMBL. So we are hoping this is useful to the community. From natural products to chemical probes, I think that's another really emerging topic. So as you will all know, chemical probes are quite special. So they're highly selective tool molecules that help you understand the biology of a target. And we have been, or we are still part of an IMI project called EU Be Open, a 5-year project that will now come to its end next year, and where the major aim was to, on one hand, create or synthesize new high-quality open access chemical probes, and they should target also novel target families like solute carriers or E3 ligases, but then also establish a chemogenomics library. And ChEMBL was chosen as the data base to host this data. And so we did. But then in addition, we worked on creating a specialized, more bespoke resource to reflect this data, which we call the EOB Open Gateway. And so the way the data is flowing, so from an internal database called Scarab, it goes into the ChEMBL interface and then via Elasticsearch to EOB Open, to the gateway. And apart from this, we also then started flagging chemical probes in ChEMBL by annotating probes from chemicalprobes.org, but there we applied a filter for also high quality probes. The reason I mentioned this project in particular is because this project is or was and is quite challenging because of the complexity of the data that they wanted us to host in ChEMBL. So if you think about cell viability data that is measured at various concentrations, multiple time points, this is quite complicated to store in a relational database like ChEMBL. And then also you might have some visualizations, so these can't really be stored in ChEMBL. So our solution was to host the key data in ChEMBL and then have some more visualizations on the EOB Open Gateway, as you can see on the right-hand side. And then for other data like images, we link out to other databases like the Bioimage Archive, which is also an EBI, an open EBI data resource. But in order to store such data in ChEMBL, you also need to build the infrastructure. And I have to say we have this opportunity because already a few years ago, so with ChEMBL 24, these infrastructure so developments have been made. So there has been an activity properties table, for instance, included, and the assay parameters table as well. So these are metadata tables that allow you to store additional information so that you don't have to split that in multiple assays, but you can group this complex data all together. But so this leads me to talk about data curation. So as we store this really complex information, of course, this also means we need to curate more information, right? The more metadata we get, the more curation we have to do. And this is a slide I usually would show in the beginning of such a talk, but I think it's a nice picture to show now, because on one hand, it nicely shows you the different aspects of data curation we do in ChEMBL. So we standardize chemical compounds. So this is all published, and you can find the workflow we are using in a paper in Journal of Cheminformatics. Also, the ChEMBL structure pipeline is fully open on GitHub. We also curate and standardize the bioactivity data. We do some unit standardization, outlier flagging, and so on. We calculate the p-ChEMBL value. But most of the time we spend on curating assay information, and I think that's probably most relevant to this workshop. What does this mean? So that means on one hand, you need to assign your target. And it's tempting to think that the target in an assay is always a single protein, which certainly isn't true. So in most cases, it's actually not a single protein. Often it's either a protein complex, a protein family, or it's even something that is not a protein, like a cell line, a tissue, or a whole organism. So we need to assign the target, and then we link to all types of ontologies. And this is actually what we what uses up most of the time we spend during a release cycle or a curation cycle. And so the remaining minutes I want to spend by talking about projects we're working on to improve exactly this biocuration part in ChEMBL. And I also want to make a point by saying we are quite aware of all the different errors that one can make when using our data, or even errors that are being introduced beforehand. So on one hand, if you look at this figure in the left corner at the bottom here, there can be mistakes in journal articles. We can't do anything about this. Then our data extractors might do mistakes. And then during our in-house curation, we might also misidentify, do some erroneous mapping. So we can't really influence what is happening before the data reaches us. But what we can do is we can improve our data checks, and that's what we are doing now. And we can also inform the community about what we think is best practice to use our data and to clean up our data. And there has been a recent article by Greg Landrum that also discusses this, and it was also helpful to discuss this with him. So we are now working on raising the awareness of this, of the ChEMBL data structure and the annotations, and also want to, at some point, release our data check notebooks to the community and make some more recommendations. So because the reason I mentioned this is that you need to be aware that it really depends on your use case, how much you want to refine your data set, and then what you would get out of it when you use your data for modeling purpose, right? So here's one example. You can collect all the IC50 data for ABL1, or you can further filter only for the human target, or you can go even deeper and just consider wild type ABL1, and so on. So these are all questions you need to ask yourself before using our data. And we can't, of course, take responsibility for how you are using our data, but we think we are in a position to shortly also release some more recommendations. I don't have time to go into a lot of detail here, but we do a lot of data checks for different aspects of the data. And just to end this talk, I wanted to mention two more things. So protein variant annotations in ChEMBL, that's maybe something that is not so well known. So we have been introducing a variant sequences table already in ChEMBL 27. So this information is available, but it's not information you will find at the target level, but at the assay level. So it's some information you find in the assays table. So you need to be aware when you combine data from different targets, there might you have to consider the protein variant annotations. We lately also worked together with Gerard van Besten's lab to update a bit our data variant extraction pipeline. So they did it in a slightly different way than we did it. And together we could just enhance these annotations. And this has been published on ChemArchive. So just have a look at the work we did here. That's my final slide, and probably for me, the most important one, and it's unfortunate I can't talk longer about this. So this is also unpublished content, but it's just interesting. 10 years ago, when I've been a user of ChEMBL data, I was already thinking, okay, now we have all this amazing data from ChEMBL, but how do we know which assays we can combine for building machine learning models? And what I'm trying to do now with the team is improving assay annotations. So we do already map the bio format. These are things like cell-based format, tissue-based format, and the endpoint, this is the readout, like IC50, Ki. What we are not doing yet is, for instance, annotating the experimental method. So we developed some models, NLP models, to now map that as well, also identify the method in the assay description and then map it to the bio-assay ontology. So this is work in progress, but I'm hoping we can release this information next year. And we're also in the course of writing a paper about this. I think my time is over. but I'm also coming to an end. I want to thank the whole team. So it's a large team, around 25 people that are maintaining our 4 resources. And these people I listed here, the people involved in the project I've presented, and then also some former group members who have been part of these projects, and some members of Gerard van Westen's lab that were working with us on the variant pipeline. And last but not least, I want to say the cake is only half eaten, so I think there are still a lot of things to do, and I'm looking forward to an exciting future. Thank you.
Thank you so much, Barbara, for a wonderful presentation. I hear your thoughts about the importance of assay annotation, and hopefully maybe we'll bring you back to a webinar or something and you can talk more about it because we'd love to hear that. So we have multiple questions for you. So we'll start taking questions. Here's one. So how do you ensure that ChEMBL captures assay conditions in sufficient details? And the second part of the question is, how is it ensured that 2 IC50 Ki values are replicates of the same experiment for a molecule?
Okay, so the first part, how do we make sure that we have all the necessary conditions? So the roughly negative answer is we can't, right? So we have to basically rely on what the depositors would give us. What we do have is a manual that describes the kind of information we need if data is being deposited to ChEMBL, but we are quite flexible in terms of how an assay, for instance, is being described in the assay description, and we need to keep that flexibility. That's what I think, because otherwise you can't really press everything in the same schema. So to ensure that the necessary conditions are delivered to us, we can just use our understanding of biology to ask the right questions. But it might be that sometimes we are missing some of the metadata. So we can't guarantee this. So in that case, the depositor takes the responsibilities. For papers, so medchem literature, we are relying on our data extractors to extract that information. They now have 15 years of experience to work with us. They use always the same format and same structure, so I'm pretty confident that they would extract everything we need, but we can't say with 100%. And the second one, I don't remember now. Could you please read it out again?
Yes. So how is it is ensured that 2 IC50 Ki values are replicates of the same experiment for a molecule?
Well, I'm not sure I understand. So usually, most of the data we don't report replicates at all. So I'm not sure I understand that question. Usually we would just for one assay, one compound target pair, get one measurement. If people would want us to store replicates, that's part of the metadata we would store. I'm not sure.
Thank you, Barbara.
Yeah.
So please, then one more question. Yeah. Go ahead, Barbara.
No, I just wanted to say, please write in the chat and I can answer later if I misunderstood that question.
Fantastic. So another question here is, in ChEMBL, different variants of the same parent protein are assigned the same target ID. What measures are being taken to address this issue?
So the measures that are taken is conversations we have. So we think the information we give is enough to address the issue of identifying variants. We could introduce a new target identifier for every variant. The question is it necessary? Just the way we store it is maybe not so convenient for everyone, but all the information is there. So we are in discussions if we want to change that or not. It would just request us to go back over a lot of legacy data, like 14,000 targets to address this. So it's more an issue of having the funds to do that. So we might do it or not, but just be assured the variant information is there. It's just stored at the assay level.
Great. Thank you so much, Barbara. There's so many more questions. So if you could just type some of these answers, that would be fantastic. And we'll move on to our next speaker. Appreciate your time. Thank you so much.
Thank
All
you.
right.